Hi everyone, and welcome to my A-level biology lecture series. In this lesson, we will explore adenosine triphosphate, or in short, ATP. At the end of this lesson, you will be able to describe the features of ATP that make it suitable as the universal energy currency. State the ATP is synthesized by transfer of phosphate in substrate link reaction, as well as the chemiosmosis in membrane of mitochondria and chloroplast. ATP, adenosine triphosphate. So a molecule of adenosine triphosphate, ATP, is a phosphorylated nucleotide because normal nucleotide consists of nitrogenous base, one pentose sugar, as well as one phosphate group. However, adenosine triphosphate or ATP composed of adenine, one of these nitrogenous bases, ribose, a type of pentose sugar, okay, the pentose sugar with 5 carbons, okay, sugar, as well as 3 phosphate group. So this made adenosine triphosphate different from the normal nucleotide, which consists of 1 phosphate group only. So the diagram shows us a complete structure of a molecule of ATP. Please bear in mind that ATP is not a form of energy, but ATP serves as the energy currency in all living organisms, providing an immediate source of energy for cellular activities. So, how ATP can act as universal energy currency? Whenever we use the word universal, means no exceptional. So therefore, ATP is used in all living organisms, shows that ATP is the universal energy currency. In terms of the structure, ATP is very small, okay, compared to those macromolecules such as polysaccharides, proteins, polynucleotides. They are very, very small. And most importantly, they are water soluble. Being small and water soluble allowed them to be readily transported within the cytoplasm, from one part of cytoplasm to another part of cytoplasm within the cells. Okay, so for example, it can be transported from the region near to a mitochondrion, okay, been transported to another regions of the cells, for example, where the synthesis of polypeptide take place, okay? And the most important thing here, the structure of ATP allowed them to serve as an intermediary molecule between these two reactions, energy releasing and energy requiring reaction. So we have these two types of reaction. First, we have catabolic reaction where energy is released. For example, oxidations of carbohydrate, fat, and protein. So these processes release energy. And we have another type of reaction which require energy, or we call them as ATP requiring process. For example, synthesis of the large carbon compound like protein, DNA, carbohydrate. So we have these two types of reaction, energy releasing and ATP requiring process. So ATP is a suitable molecule to become the intermediate molecule between these two processes. So when energy is released through catabolic reaction, so this energy will be used by ADP. ADP stands for adenosine diphosphate. Okay, so ADP react with phosphate group. Pi stands for inorganic phosphate group. So absorb the energy. So once the energy is absorbed by ADP, together with inorganic phosphate group, ATP is formed, is produced. So therefore, we can capture the energy released by the catabolic reaction. On the other hand, when anabolic reactions take place, energy is required. So therefore, hydrolysis of ATP releases energy for this anabolic reaction. So ATP is a suitable molecule to be used to act as the intermediary molecule between the energy releasing and energy requiring reaction in a cell. Zooming in, okay, so if you look at the reactions here, we have ADP and ATP. So ADP with two phosphate groups. You look at the diagram here. Okay, we have two phosphate groups here. Can you see that? Okay, so these two phosphate groups, so therefore with these two phosphate group, so this molecule is known as ADP, adenosine diphosphate. So during energy releasing process, okay, so energy is released. So this ADP combined with phosphate group to form ATP. So during this process, it's a form of condensation. So water molecule is released. On the other hand, when energy is required, hydrolysis of ATP with this water molecule add in into this reaction. So ATP is hydrolyzed. When ATP is hydrolyzed, a phosphate group is removed and the energy is released. So this energy can be used for the anabolic reactions as well as those energy requiring process. So ATP synthesis occurs when energy from the respiration, for example, glucose oxidation, is used to phosphorylate ADP. So the word phosphorylations take place here involve the transferring of the phosphate group to the ADP, forming ATP. So the entire reaction in this case shows that it is a condensation process. So ADP react with inorganic phosphate group. With the supplies of the energy from the energy releasing process, ATP is formed and water molecule is released. So this is an example of the condensation reaction. Okay? So the reaction stores this energy in the high energy phosphate bonds of ATP. On the other hand, ATP hydrolysis breaks down the ATP into ADP and inorganic phosphate group. And the enzyme that catalyzes these reactions is known as ATPase, which lower the activation energy required for the hydrolysis. When one inorganic phosphate group is removed from the ATP through the hydrolysis process, 30.5 kilojoule per mole of ATP, also of energy, is released, which can be used for the cellular processes such as active transport, biosynthesis, as well as muscle contraction. And the interconversions between the ATP and the ADP is very, very high. So we say that ATP has a high turnover rate. It can be rapidly hydrolyzed and resynthesized within the cells to ensure a continuous supply of energy when this ATP is hydrolyzed. So and what makes this process high turnover rate? One of the properties here is the interconversion between the ADP and ADP is a single step process. So therefore, it is efficient for the immediate energy release as well as for energy storage. So how ATP can be synthesized in eukaryotic cells? Okay, our focus is in eukaryotic cells. So there are three main processes First of all, through substrate-linked phosphorylation. So this process occurs during respiration when a phosphate group is directly transferred to ADP to form ATP. The second process or second way through oxidative phosphorylation during aerobic respiration only. Anaerobic respiration will not synthesize, will not form ATP through oxidative phosphorylation. Okay, so this process requires the energy from the electron transport chain and oxygen as the final electron acceptor. So therefore, in anaerobic respiration, oxidative phosphorylation won't occur. The third way is through photophosphorylation during photosynthesis, where the light energy drives the production of ATP in chloroplasts. Photophosphorylation will not be discussed in this topic, but will be explored in photosynthesis. We start with this substrate-linked phosphorylation. So during this process, we can see that we have the substrate here. So substrate has one phosphate group, and we have the ADP. So both of them bind to the active site of an enzyme. So substrate-linked phosphorylation actually involves the direct transfer of a phosphate group from a phosphorylated compound. In this case, PP, the substrate, is a phosphorylated compound or the substrate. So involve the direct transfer to ADP so that ATP can be formed. During the process of transferring this phosphate group, reorganization of the chemical bond here releases energy. So this energy will be captured by ADP to form ATP. Look at this diagram, you can see that the chemical bond, reorganizations of the chemical bond releases energy. So this energy will be used by ADP to combine with phosphate group to form the final product here, ATP. So this process occurs in the cytoplasm during glycolysis, as well as in the mitochondrial matrix during the Krebs cycle. Don't worry about glycolysis and Krebs cycle. We are going to explore both of these reactions in the future lesson. So a total of 4 ATP molecule can be produced per glucose molecule. In glycolysis, we will have a net gain of 2 ATP, and another 2 ATP are produced from the Krebs cycle during this substrate level phosphorylation or substrate linked phosphorylation. The next way or process that produce ATP is through oxidative phosphorylation. So oxidative phosphorylation only can occur during aerobic respiration where oxygen must present. So during this oxidative phosphorylation, due to a series of reactions, there will be accumulations of hydrogen ions or proton at the intermembrane space here. So therefore, high concentration of proton in the intermembrane space, low concentration of proton at this mitochondrial matrix. So a steep proton gradient or electrochemical gradient is generated. So for protons to move back from intermembrane space back to the mitochondrial matrix, impossible for protons to diffuse across the phospholipids bilayer here, the inner mitochondrial membrane, because proton is positively charged. So impossible for protons to pass through or diffuse across the inner mitochondrial membrane. The only way for protons to pass through this, to cross through the inner mitochondrial membrane is through the proton channel, which is associated with ATP synthase. So ATP synthase is an enzyme, okay, to catalyze the reaction, the phosphorylations of ADP to form ATP. So how this can be achieved? So first of all, the proton diffuse across this proton channel. When this proton diffuse across this proton channel, energy is released, okay? High potential energy, low potential energy. So energy is released. So this energy will be captured by ADP to form ATP. So entire process here is known as the chemiosmosis. Similar process occurs in the chloroplast, okay, where we are going to explore this in the next, right, in the future chapter, photosynthesis. Oxidative phosphorylation occurs in the mitochondria only, during aerobic respiration. So this means that this reaction actually depends on oxygen, which act as final electron acceptor. So during this oxidative phosphorylation, a series of reactions actually take place in the inner mitochondrial membrane. First of all, the energy from the electron transport chain is used to pump the proton. First of all, pump the proton from the mitochondrial matrix into the intermembrane space. By doing so, a steep proton's gradient, which is also known as electrochemical gradient, is created. Okay, so what is meant by electrochemical gradient? Electrochemical gradient is due to the charge distribution, because when this energy released through this electron transport chain pumps the protons from the mitochondrial matrix into the intermembrane space, so there's an accumulation of proton in the mitochondria intermembrane space. There's a lot of positively charged ions, proton, at the intermembrane space compared to the mitochondrial matrix. So this kind of gradient is known as electrochemical gradient. And protons move back into the mitochondrial matrix by the process known as vacillated diffusion through a proton channel. And this proton channel is associated with ATP synthase. Okay, so as it's the vacillated diffusion, so the process is down the proton's gradient. So ATP synthase, okay, will capture the energy released by this proton, as we know that from the high potential energy to low potential energy. So this energy is used, okay, to phosphorylate ADP into ADP. And to produce one ATP molecule, it requires the movement of 3 protons, okay, in the synthesis of one molecule of ATP. With that, we have come to the end of this lesson. I hope you have found this lecture series helpful and informative. If you have any questions, feel free to leave them in the comment section. I look forward to seeing you in the next lesson. Have a great day.